Welcome to part 3 of a 3-part lecture on microbial pathogenicity. We in this we just talked about exotoxins, finished up part 2 discussing exotoxins, and now we'll begin by looking at the second major category of toxins called endotoxins. So as with exotoxins, exotoxins exit out of the cell, they come from the cell. Endotoxins come from within the cell, or they're part of the cell. And they happen to be part of the cell walls, the outer portion of gram-negative cell walls. And they are lipopolysaccharides, so basically fatty carbohydrates. And they are released whenever the cell wall lyses. And I put in parentheses there antibiotics because that's what antibiotics do. They cause the lysis of bacterial cells, and as a result of all of these, say, millions of bacterial cells that are lysing because of taking antibiotics, a lot of endotoxins get released as a result of taking antibiotics. And so sometimes when you're on an antibiotic therapy, you don't feel great and you're taking your medication. You think, well, why isn't it working? What's going on? Well, the reason why is because you have to give the antibiotics time to reduce the number of bacterial cells, and at the same time, if it's a gram-negative bacterial infection, you have to wait for the endotoxins to dissipate to levels where you don't feel so sick anymore. And so as a result of that, the result of the antibiotics lysing all of these bacterial cells, endotoxins are getting released, and then you don't feel so good as a result of that. So people expect feeling better immediately after beginning their round of antibiotics when it depends, of course, what it is that's infecting you. And it doesn't always turn out this way either, because antibiotics, of course, as you know, are non-selective. If it's a bacterial cell, they kill them. But in some cases, if it is a large number of gram-negative bacteria within you that is causing an infection, sometimes you don't feel great from the start. And so symptoms can persist, and you think, well, why? And that's the reason. It's because the cells are lysing, releasing their endotoxins, and the illness seems to persist. Endotoxins stimulate macrophages to release large amounts, and sometimes even toxic levels of cytokines. What do they cause within us? What happens to us with endotoxins? And well, fever, chills, weakness, aches, blood clots, miscarriage, shock, death. So it could be any combination of those as well that are causing illness within us, and all of these signs and symptoms are coming together to make us feel awful. In the United States, just specifically septic shock, 750,000 cases within the United States. Within a month of the 750, 250,000 die in a month. And then about total 375,000 die within 6 months. So you can see the mortality of endotoxins and their effects they're devastating. So they are deadly compounds. This just shows where they kind of are. I mean, it's of course not reality, but it's in the outer membrane of gram-negative bacteria. So if you look at the structure of gram-negative, you have the outer membrane, which gram-positive bacteria do not have. Then they have a much thinner layer of peptidoglycan in their cell wall compared to gram-positive. And then they have the inner cell membrane, the plasma membrane, and then within there would be the cytoplasm. So that's why we say endotoxins gram-negative, because they're part of the outer membrane, which gram-positive bacteria do not possess. And this picture is showing specifically it's those lipopolysaccharides. And they are embedded, they're incorporated into the outer membrane, and so they're not just sloughing these things off randomly. They are liberated as a result of lysis of the cell. So when the cell bursts, these lipopolysaccharide endotoxins get released. So very simple graphic here. No, it is not a NASCAR racetrack that has gone awry. It is supposed to be the endotoxins leaving the outer membrane of the bacterial cell, the gram-negative bacterial cell. So once that outer membrane breaks apart, the endotoxins, they take a trip and they leave, and then they create all their problems and issues within the host that we just discussed. So this chart is important, and as I said in part two regarding exotoxins, if you were a student of mine, I would really want you to know this chart because it summarizes it. So source, gram-negative. Is it a metabolic product? No. They're present in the lipopolysaccharides of the outer membrane, so it's not being released by their metabolism. It's just being released from the cell itself. What is it made of? Lipopolysaccharide. That's fun to say. Say lipopolysaccharide. Does it induce fever? Yes, they do. Are they neutralized by an antitoxin? No. And their LD50 is relatively large. So you can see these are, in some cases, at least with the last three, certainly with fever and neutralized by an antitoxin and LD50, they're the exotoxins are the exact opposite with those three characteristics. So this is okay, fever is caused by a number of issues. So I don't want you to think when you look at this slide, this is the only way that fever is caused. No, what I'm looking at specifically here is endotoxins and their ability to cause fever. And again, this is a simplified graphic, of course, but it gets at the major events that's happening. So the thing on the left there, that's the purple thing, that's supposed to be a macrophage, which, I don't know, to me kind of looks like a dinosaur's skull or its head or something. But anyway, that's a macrophage ingesting a bacterium. And this is a gram-negative bacterium. As a result of the breakdown of the bacterium, endotoxins are released. When those endotoxins get released, that induces the macrophage to produce interleukin 1. Interleukin 1, which is in a group of proteins called, they have immunomodulatory functions. And there are, as you can tell, obviously, if it's interleukin 1, there must be other interleukins, and there are. But in this particular case, we're looking at how it's related to fever, which interleukin 1 is involved in other bodily processes besides fever. But back to the graphic, interleukin 1 is released by the macrophage into the bloodstream. This interleukin 1 then travels to the hypothalamus in the brain. Then, as many things are in the human body and within an organism, it's like a chain reaction. One thing happens, then the next thing happens, then the next thing happens. So it's a cascade of events, the falling of dominoes, whatever you want to call it. As a result of the IL-1, that IL-1 induces the hypothalamus to produce these proteins called prostaglandins, which reset the body's thermostat to a higher temperature, producing a fever. And this is a natural process. And fever is a beneficial process to us in a number of ways. So when you have a fever, let it go for a while. I mean, you don't want to get to the point where it's, so debilitating that you can't stand it any longer, but allow the fever to do its job because it does have beneficial effects, such as increased white blood cell production, reduction in the number of bacteria by slowing the reproductive rate, and so on. There are other valuable benefits to a fever. But this is just looking at a snippet of it and one example of how fever is caused by endotoxins. So that ends pathogenic effects and pathogenicity of bacteria. Let's look at viruses. And this part finishes out the chapter rather quickly. So bacteria, they're the star of the show in this chapter in terms of the pathogenicity. And now we'll look at the other categories of microbes. So what cytopathic effects do viruses cause? Well, first let's look at the term cytopathic. Cyte means cell. Pathic means bad things happening. So these would be bad things, destructive things happening to cells. They can lead to they can stop the synthesis of macromolecules, proteins, nucleic acids, carbs, and lipids. They can cause lysosomes to release enzymes. That'll kill the cell. So when they do that, they utilize the lysosomes to break the cell open and then liberate the viruses that are waiting to get out. And so they are capable of using the lysosomes to induce early apoptosis. And then the cell breaks apart and the viruses then get liberated, and then they can go out and infect other cells. They form inclusion bodies within their host cells. What are they? They're viral parts in the host cell cytoplasm or nucleus, and we can actually use them to identify a virus because a number of viruses have very specific inclusion bodies that only those viruses produce. And so it becomes useful in identification. They can form giant multinucleated cells, which then means more DNA available to the viruses, which is what they're after. They want to use our DNA so that they can use our code to make proteins that they need. And so they induce the cell to become multinucleated and become a massive cell. They interfere with immunity. They cause the production of interferons, that's actually a positive protective effect. Interferons are produced whenever a host cell is say under attack by viruses, and so these interferons get released from the host cell, which then signal to neighboring cells that viruses are coming, prepare for the attack, and then what the neighboring cells can do is produce antiviral proteins that will prevent the replication of viruses when those viruses enter the neighboring cells. They can cause an autoimmune response as well, which can lead to a whole host of problems within a person, depending on the person's genetics and if are they susceptible to developing this autoimmune response. Number of viruses can do that. They inhibit contact inhibition, which then will lead to piling up of cells. So many of our cells, whenever they contact each other, they stop dividing. They don't pile up on each other. Well, what viruses say is, heck with that. We want to inhibit this. We want to get as many cells as possible piling up so that they can then just move from one cell to another very quickly, just going from one membrane to the adjacent membrane right next to it and infecting the cells there as well. This, of course, then can lead to tumors and oncoviruses, and then you end up developing a type of potentially developing a type of cancer as a result of these oncoviruses that are infecting us. So that's viruses and their cytopathic effects. What do fungi do? Fungi, they have waste products and they can cause symptoms as well. And there's a number of symptoms that they can cause, from something that's mild to something that can kill you. They can provoke an allergic response, and many people are allergic, of course, to fungal spores, for example, that are produced by, say, penicillium or rhizopus or aspergillus, and so those spores then can create usually respiratory distress for people because of the allergic response that is provoked by these spores. Fungal toxins, which by the way are called mycotoxins, and so anytime you see MYC, it's referring to fungi. And so if you want to be a real fungi, you become a mycologist. Anyway, what these mycotoxins do is they inhibit protein synthesis. They can cause systemic vasoconstriction, and because it's systemic, it can lead to gangrene, especially in the extremities, of course, leading to the death and destruction of tissue because they're not receiving blood supply. They cause hallucinations, which some people voluntarily go after. And so these mycotoxins can become somewhat like a neurotoxin and causing hallucinations. They can cause cancer and death as well. Some, of course, with many mushrooms, for example, there are some that are deadly because their mycotoxins are so potent. They're so toxic to us. Some produce proteases, which alter the membrane to allow fungal attachment as well to the membrane, so that they can then attach to the cell. And then they have a place to begin to replicate. They have a very thick capsule, and that capsule can prevent phagocytosis by macrophages. Because of the thickness of the capsule, the enzymes are completely ineffective against it. Protozoa, they have a number of pathogenic properties, and protozoa are the category of these microbes, and probably number one that I'm sure you've heard of, malaria, for example. Well, what is one specific pathogenic property? Well, they can actually two. Sorry, they grow in phagocytes. The main one, and why I said one, that's the one that I wanted to highlight, was antigenic variation, and so what I have here in this example is supposed to be what are called trypanosomes, and this is specifically with a particular protozoan disease called African sleeping sickness or African trypanosomiasis. And so what happens with these protozoan and this protozoan disease is you develop resistance against a particular clone of the protozoan, and that clone is called a trypanosome. And then those numbers become huge. And then you begin to develop an immune response against it. Well, that clone then disappears and puts on a new coat, in this case a green coat. So it switches from a red coat to a green coat. Now it becomes clone B. And as far as our immune system is concerned, that's a brand new infection. Then clone B reaches a peak in its numbers, and then it starts to decline. And then out pops clone C with the blue coat, and so on. And this just keeps happening again and again and again, and it just exhausts the immune system. So just when you start developing antibodies against a particular clone, that clone's gone, and a new clone has come on the scene, and then you have to develop antibodies against that clone, and then that clone is gone, and a new clone appears. And so this is what it's a classic example of antigenic variation. So they're causing a variation of their antigens or their surface molecules, usually proteins, on their surface. And so every time a new antigen comes in on their surface, it's like a brand new infection that is occurring within an individual. It's relentless. It just progresses on, and it just wears a person down to the point of death. So a major pathogenic property of protozoa. What do helminths do? Well, helminths are parasitic worms. Well, they use our tissue. They use the host tissue for their own growth. And so they're parasitic as a result of it. The presence interferes with our host function, and their metabolic waste can cause symptoms. Now, helminths are not, as mature adults, microscopic, but they go through microscopic stages in their life cycle. So that's why you look at and study helminths in microbiology. Many of them, in many cases, they aren't that bad. They cause symptoms, and in some cases, like with a human tapeworm, for example, sometimes there are no symptoms until you start excreting segments of the tapeworm out, and you realize then you have a tapeworm infection, which would be quite an alarming revelation, I would think. But these helminths, they create a huge number of infections worldwide. In fact, these parasitic worms, around almost half the world's population are infected with parasitic worms, helminthic infections. About 3 billion people are currently, at this time, infected with a parasitic worm of some type, where you can see the samples that I have there. Some of them, their pathogenic properties are devastating, and some of them not really at all. So anyway, I just wanted to mention that just to cover the helminths. Last group we'll look at will be algae. So what do algae do? Well, they create a number of things. I want to highlight this one here, though. What pathogenic properties do they they produce these toxins called neurotoxins by a group of algae called dinoflagellates, or otherwise known as plankton. Um, what are some of the symptoms that are seen by these dinoflagellates? Paralytic, diarrheic, and amnesic shellfish poisoning. And the effects of these three different types of shellfish poisoning are similar to botulism. And so you see the picture on the left, the micrograph of this tiny little dinoflagellate, and they possess this a number of pigments, one of which is red or kind of a brownish reddish pigment. And whenever their numbers increase to epic proportions, you develop a red tide, as you can see in the picture on the right. The number of dinoflagellates in there would be astronomical. It would be a colossal number of dinoflagellates to make a red tide of that magnitude. What causes these? There are a number of factors that come together. Low salinity of the water, high nutrient concentration in the water, higher overall temperature, and then this just leads to what's called an algal bloom, an explosion of dinoflagellates. Um, what happens here with the shellfish poisoning? So you have shellfish, say like clams, oysters, scallops. They're filter feeders. And what are they filtering for? Plankton, the dinoflagellates. And if there is an abundance of them present, they then will accumulate these dinoflagellates and their toxins into their muscular tissue. And then people consume these shellfish, and as a result, and often undercooked shellfish, like raw oysters, for example, they will develop these diseases, these shellfish poisoning diseases. Um, these red tides create an enormous loss of aquatic life, from fish to birds to mollusks. Millions of organisms will die as a result of these red tides, because the toxins are toxic not to just humans, but to other life forms as well. Um, and the toxins, even if the red tide is concentrated enough, because of the wave action and the aerosol spray of the water, the toxins can then be liberated into the air, and people can breathe these toxins in, which can lead to respiratory illness as well. So, anytime red tide is near, stay away from the red tide. And not only do not go into the water, obviously, don't go near the water because of the aerosol transmission of these toxins being released into the air. Portals of exit. So these microbes get in through portals of entry, so there must be a portal of exit. So how do they get out of us? Respiratory tract. This is how, like for example, SARS-CoV-2 gets released through coughing, sneezing, breathing, talking, laughing. So they are released into the air through droplet transmission. As you can see in this picture of this gentleman sneezing, look at the droplets. Tens of thousands of droplets getting released, carrying viruses with them. Gastrointestinal tract through feces, or spitting them out in saliva, being released by saliva. Say, for example, in rabies, for example, when the rabies virus get into the salivary glands, they get released into the environment through the excessive salivation that's caused by having a rabies infection. Genitourinary tract through urine, penile, and vaginal secretions, so they can get liberated into the environment, passed on to others that way. Through the skin and through the blood. Biting arthropods. I mean, this is how these diseases get spread. African sleeping sickness and malaria, Chagas's disease, they get spread through an environment through these biting arthropods, picking them up from a person who is actually carrying these pathogens and then passing them on to the next host. Through needles and syringes, they get passed on as well. So these are all portals of exit. How do they leave their host? And this is just a graphic showing and summarizing quickly summarizing what some of this chapter focused on. Beginning with the portals of entry, how do they get in? and then ways in which they penetrate or evade our defenses, and then after they have done that, what damage do they do, cytopathic effects, and then how do they get out of their host with the portals of exit. Well, that's chapter 15, Microbial Pathogenicity. That was the end of part 3 of this 3-part lecture. I hope that was educational and beneficial for you. And before I go, I just want to remind you to subscribe to Tink Talks, like this video, leave a comment, much appreciated, and share as well. If you receive benefit from this, then maybe somebody else would as well, and that's my goal. So thank you for watching.